I'm going to talk about clinical utility of whole genome sequencing. And it's a pretty interesting topic, especially taking into account that we were promised that that will be the future for quite a while already. And if we look at profile of the company that I currently represent, you will see why it's, for us, it's particularly applicable. So VarI is a company that provides clinical genomic services, and we are starting from the actual collection of saliva or blood sample and going all the way into production of clinical reporting when the interpretation is done either by our clinical geneticist or with collaborative diagnosis by ordering physician with our clinical coordinators. So we are dealing with clinical analysis of exomes and genomes every day, and we have gathered quite a lot of data to be basically judging in this topic. So this is a picture from a 5-year-old Nature paper that projects that we will have actual clinical implementation of whole genome data somewhere in 2040s for personalized medicine. Do we really have to wait this long? Let's see. So to analyze this, we have to look what is current standard of care in clinical genetics, and if the actual clinical grade whole genome sequencing is a legitimate alternative. And we have to look at two things. Basically, it's cost and the detection power of these techniques. So first, let's look at the alternatives. And in the recent year, just recent year, there are few publications that looked at implementation of whole exome sequencing. Nobody were looking into whole genome yet. But comparing whole exome sequencing with the actual standard of care right now. So there are a number of papers. I don't have right now time to go over this. But the conclusion is that if we compare it to current standard of care, which is typically would be Fragile X or ACGH, then some panels, either Sanger sequence or next generation sequencing data. And we will see that that's drawn from a combination of all these papers, that traditional testing costs much more per person and takes tremendous amount of time. Why it takes so much time? Look at the cases, if particularly is cases of mild phenotype, when you have mild dysmorphologists or just intellectual disabilities, and an early age of these patients, which are rare disease diagnosis patients, you can't really point at a particular suspicious set of genes or to look into particular disease. So as one of the prominent dysmorphologists said once, you should give them time to grow into their syndrome. So that means that these children and these families, they suffer for years until they develop the symptoms that allow to point the diagnostics into the right direction. So all these could be avoided if we look in directly into whole exome or even better in whole genome sequencing. And this is just an example of a number of tests from one of these publications that these patients undergo until they finally went to whole exome or whole genome sequencing. And with this graph that you obviously know already, you see the cost of whole genome is not that much different right now from the cost of whole exome. And we're talking about clinical grade sequencing. It's not research grade. It's actually clinical grade in clear, HIPAA compliant environments and so on and so forth, validated. And if we're looking even into trio analysis, the whole difference would be of $1,500, which is already comparable to the prices of basically just another gene panel that you do. But what are the benefits that you get from whole exome compared to whole genome? I will not even talk about genome versus panels. It's obvious, yes, but should we opt to save these $1,500 and go to exome or go directly to whole genome? And there are three main differences and advantages, I would say, in whole genome sequences compared to exome. First would be better coverage in its own territory in coding regions. Then, of course, regulatory regions and phasing that you can't get with exomes, and structural variants. And not only structural variants, large structural variants that are detectable via ACGH, but also smaller copy number variants that you can't detect with basically any other technique. So let's start first of all with coverage. You can see that that's obvious statement, of course, that typical exome covers 50-60 million base pairs, when whole genome covers 98% of the whole genome. And if we look just into disease-causing mutations, if you typical whole exome sequencing will not have coverage to see any mutations in approximately 15,000 known pathological mutations. That's quite a lot, compared to just 500 in whole genome. That just by the regions. Of course, regulatory regions, right now we already have enough annotation to actually look into these regions and to understand what this stuff means, both annotations and population frequency. Particularly with publishing of GNOMED, 2 months ago, no, 3 months ago already, which provides population frequency from 30,000 whole genomes that allows us to truly look into conserved region on whole genome level. When we had just exact data, we could not leverage this information. So basically, anything outside of known coding regions was considered unknown territory, and you couldn't really judge the importance of the variant. Exon coverage, yes. So even if we look just into coding regions, because of the uniformity of reads by whole genome sequencing, it provides better coverage even on the coding region. So this is a comparison of 100x, typical 100x exome, and 30x whole genome. So even with lower coverage, we see that the numbers where the depth of the read is below detection value, which we consider 8 reads to call something, we are missing more than 2% of pathological variants on coding regions in exome compared to 0.3% in whole genome. Not big numbers, but if you actually have patients that have these mutations and you can't provide the answer for them, it means something. So just example of this particular situation is it's one of the case study. I will not go into the details of the phenotype, but we were able to detect with whole genome sequencing this small deletion that was not readily detected by exome, despite the fact that it's located in well-known gene and that is associated with CHARGE syndrome, and we were able to get diagnosis for this particular patient. Now, probably the main advantage from my perspective is structural variants. Because if you look into coding region, you probably, if you go instead of 100x with exome, you go to 200, you go to, I don't know, 500 you probably can get to same coverage of mutations as you have in whole genome. But what happens with other things that you just can't physically detect with whole exome? And structural variants is one of the things. So whole genome provides an opportunity to actually have one test that covers all genetic aberrations. So you don't have to do RACGH, which is done routinely and clinically, and also has its resolution limits. How do we do at Variandix detection of structural variants? We have developed a pipeline that has three major steps. First of all, would be the detection of the variants. There are two main techniques, breakpoint analysis and rep depth analysis. So what we do actually both of them, and then we combine the information from both of them and look how fit the variants are. Then the variants that we find, we do annotation to them, and the annotation, it could be tricky. Because if you look at the HGMD, for example, which is right now the biggest source for human-created pathological annotation for disease-causing mutations, you will see that there are plenty of structural variants there, that are about 20,000 of them. There are deletions, well annotated, insertions, duplications, rearrangements, but they are not mapped. They don't have genomic coordinates. So our system can take this and to convert them into genomic coordinates so we can annotate with them. So in addition to these unmapped copy number variants, we do have also we are looking into overlapping point mutations. So if there are known point mutations or small deletions in insertions in particular pathological genes, we want to know it as well. And then we do, as a last step, we do parallel filtering, and that allows us to reduce the number of candidate variants from few hundreds, that would be the typical result in analysis of single whole genome for structural variants, to just a few that could be easily looked at and sorted by physicians who are not bioinformatics professionals, and they don't want really to know all these parameters. And these filters include zygosity, they look into parameters of call quality, population allele frequency from DGV and other things, look into phenotypes. But what I mean by parallel filtering is that every type of aberration, which is, for example, homozygous deletion versus heterozygous deletion, or duplication that is called just by DEPs, compared to duplication that is called by breakpoint analysis. So they go through different filter settings. So at the end, the picture is combined, and I will show you an example of, again, one study that basically gives you a typical example of what you see. So this study is a person with muscle disease and muscle weakness, and their exome was tried and multiple panels, nothing was found. And we see at the results, and that's a type of the results that we get typically. You can see a list of these variants. There are plenty of them, but they are actually grouped by parameters that we used for filtering. In this case, you can probably see here, we have associated phenotype pipeline. So these check marks here on the 4 variants means that there are known structural variants in the literature that overlapped with these variants that are called here. And here it means that there are also some variants that have point mutations that are overlapping with these features. So we can quickly look at other parameters here, and we see, for example, that we don't have C-invenator overlap. That means that these are not called with read depths. So at the first glance, we can just look at those with some values here. And that would be just one variant here in the top 4 that do have some interesting annotation. And we look at this variant, just click on it, and we see that this is actually duplication in X chromosome. Again, the first thing that you probably want to do, you can go to this clip and look at IGV at the breakpoints to see if the variant is real. And we see that both breakpoints are very nice here. You can see very clearly that it is duplication here, starting right at this position. Sorry, yeah. So you see the reads, reads here, and then you can look further into what actually happens here. You see that this is a deletion of about 300,000 base pairs that involves the DMD gene, and it involves exons 3 to 9. And if you scroll down, we actually see all this annotation that we found in unmapped mutations in the GMD, that there are actual known duplications of this in this gene of different number of exons. Now you can see here, and the disease phenotypes is the actual muscular dystrophy and Duchenne-Baker dystrophy and so on. And there are multiple publications that back them up. So there are actually few more of these, another page of these. If you scroll down a little bit, we actually see that in addition to known structural variants, non-pathological structural variants, we also see some point mutations, again from HGMD and from CleanVar as well. You see that these are phenotypes, and we see that there are 160 pathological mutations, most of them associated with different types of muscular dystrophies, and slightly less in CleanVar. And again, if you scroll down further, you see that you see all these technical parameters that we are actually using in filtering that definitely you don't really want to know. It's just who has interest in this can look it up, and I can show you. But just important to know that these are parameters that are detected, and they are used in filtration and prioritization of the variants, like split treats that we saw on the breakpoints right now. So if you look at this guy in the middle, and he was pretty frustrated with the question, are we there yet? We actually can now answer to this question that yes, looks that we are there. We are able to utilize whole genome sequencing with relatively low cost. So this person, for example, was diagnosed with just a single whole genome, no need of trio in this case. And just from our experience with whole genome analysis, in the case of trios, we are approaching to 80% diagnostic yield right now, which is a pretty significant number. And the difference in cost between whole exome and whole genome already does not justify opting out of it. And I want to acknowledge particularly Greg Faust, who was one of the developers of the pipeline, and a few more people.